Then the junctional transmission. So here the junction means neuroeffector junction. The arrival of the action potential at axonal terminals initiates a series of events that trigger transmission of excitatory or inhibitory impulses across the synapse or neuroeffector junctions. So here there are 5 steps in case of the junctional transmission. So axonal conduction, you have understood now. The action potential comes, the resting membrane potential minus 70 millivolt upshoots to minus 55. Then there is inward movement of the sodium pump because of the opening up of the sodium pump. Then immediately after the attaining the depolarized state, once again the repolarization starts because of the opening up of the potassium channels. The potassium ions starts coming in, and ultimately the repolarization occurs. Once again the resting membrane potential is maintained, whereas the junctional transmission contains 5 steps. The number 1 is storage and release of the neurotransmitter. Storage and release of the neurotransmitter. The second one is combination of the neurotransmitter with post-junctional receptors and production of the post-junctional activity or potential. So first there will be a storage, then it is going to release of the neurotransmitter, and activity of the post-junctional cells initiates maybe either excitatory or inhibitory. And the third one is the initiation of post-junctional activity. The fourth one is the destruction of dissipation of the transmitter. Then non-electrogenic functions. These are the important steps you have to remember. The junctional transmission process containing 5 important steps starting from storage, release, action, then destruction, and the non-specific functions. The first one we have to discuss about the storage and release of the neurotransmitter. So usually the non-peptide or small molecule neurotransmitters are largely synthesized in the region of the axonal terminals and stored in the synaptic vesicles. So usually whenever the action potential comes, the axonal terminals, then the stored norepinephrine or the epinephrine acetylcholine is released through the synaptic vesicles. So the peptide neurotransmitters or precursor are found in large dense core vesicles which are transported down the axons from their site of synthesis in the cell body. Usually the raw materials or the precursors of the neurotransmitters are stored in the large core dense vehicles, especially in the exoplasm. And during the resting state, there is a continual slow release of isolated quantity of the transmitter. This produces electric responses at the post-junctional activity. This is called as the miniature end placed potentials, or MEPPS. Means during the resting phase, if the cell is not contracting or relaxing at that time also, there is a continual slow quantity release of the transmitter, and this produces electrical responses at the post-junctional membrane. But this is not enough to cause the action potential or the overshoot, but it makes the post synaptic receptor very active always to face any situation. That's how it is called as the miniature end plate potentials or MEPPS. So the diagrammatic representation of the miniature end plate potential is a small quantity of acetylcholine, especially in the cholinergic system, whereas the norepinephrine in the adrenergic system these are secreted from the nerve terminal in very small quantity, and each vesicle release produces weak and a plate potential about 0.5 millivolts miniature and plate potentials. A small quantity of the neurotransmitter is released which causes the miniature end plate potential. That means the end plate will be always ready to receive the stimuli or the action potential. So here the resting membrane potential is minus 70 to minus 80 millivolts. Then there is slight depolarization due to the continuous leakage of the neurotransmitters. That causes miniature and plate potential, whereas this prepares the receptors to active always. Then when action potential comes, the MEPP becomes the miniature and potential becomes the action potential because of the sodium channel opening and potassium channel closing. So whenever the action potential arises at the nerve terminals, it releases several hundred quanta of the neurotransmitter from the storage vesicles, and depolarization of the axonal terminal triggers this process, usually depolarization. So what happens is whenever the depolarization occurs, there is influx of the calcium which enters the axonal cytoplasm and in some way promotes fusion between the exoplasmic membrane and the vesicles. Vesicles are present in the nerve endings, and the depolarization means sodium shoots over. At the time depolarization, there is fusion between the exoplasmic membrane and the vesicles. So the contents of the vesicles, including the enzymes and other proteins, are discharged to the exterior by a process known as the exocytosis. Means exocytosis is a process of discharge of the materials along with the its contents. Exocytosis means outside the storage place. Then the presynaptic compartment containing the components required for the vesicle docking. So here a new term known as the docking. Docking is there, and docking means fusion of the two vesicles for the rejoining and the synapse. So for this, making the particular thing to join, synapsins and RAB3, the later a RAS family protein control trafficking and mobilization of the vesicles, and the important substances like synaptotagmin and synaptobrevin are located on the vesicle membrane, and the neurexin and syntaxin are located on the plasma membrane of the presynaptic nerve ending, and these are involved in the docking up of the vesicle. Docking means joining the vesicles, docking. So these are known as the docking complexes, the synaptotagmin, synaptobrevin, syntaxins and neurexins. And other proteins such as the synaptophysin contribute to the formation of fusion pore. So whenever the action potential comes, recovery of the transmitter is also required as in case of choline metabolite of the acetylcholine. And here you can see that the synaptic vesicle, this is a synaptic vesicle and this is a synaptic cleft. So this is a nerve ending, and all these are required for fusion between the nerve ending and also the synaptic vesicle particle as the rupture. So the important materials required for the docking complex are the synaptotagmin and synaptobrevin, and also syntaxin and SNAP5. So these are all involved in the docking complex. This entire system is known as the docking complex. So here you can see that what is a docking complex means. It's a SNAR assembly. So the trans this here what happens usually is the is transport vehicle. This uh, is going to be have a union between the tethering complex, which is containing RAB protein, and GTP, which is associated with GTP, whereas the syntaxin and T snare are two other proteins, and whereas the SEC1 and the transport vehicle are going to be fused, and ultimately there will be fusion between the two type of the vehicles with the help of the syntaxin and T snare. So the variety of the chemical substances can inhibit the neurally mediated release of the either norepinephrine or acetylcholine by interaction with receptors on the appropriate neuroterminals. Norepinephrine is able to interact with presynaptic alpha-2 adrenergic receptor. Alpha-2 adrenergic receptor is always presynaptic, and it is known as the ATO receptor, and inhibit neurally mediated release of the norepinephrine. So it's a inhibitory neurotransmitter. Whenever the norepinephrine is released, it is going to act on the presynaptic alpha-2 type of the receptor and inhibit the further release of norepinephrine. That's why it is known as the ATO receptor. Administration of alpha-2 or adrenergic receptor antagonists causes a marked increase in the release of the norepinephrine, because it is going to stimulate the release of the more and more norepinephrine. In an analog fashion, neurally mediated release of acetylcholine from cholinergic neurons is inhibited by acetyl, that's the alpha-2 adrenergic agonists. Conversely, stimulation of presynaptic beta-2, some of the beta-2 type of the receptors are also presynaptic, is associated with a modest enhancement of the norepinephrine release. Whereas all these things, adenosine, acetylcholine, dopamine, prostaglandin, and enkephalins, all inhibit neurally mediated release of the norepinephrine by means of interaction with specific presynaptic receptors. See, the adenosine receptors, the acetylcholine or cholinergic receptor, dopamine receptors, PGE type of the receptors, and enkephalins all inhibit neurally mediated norepinephrine release by one or the other mechanisms. And these receptors, especially the adenosine etc. receptors, exert their modulatory effect so by inhibiting the function of the pre-junctional calcium channels. So these are going to act through the calcium channels. So please read this particular Goodman and Gilman. These sentences are taken from the Goodman and Gilman. Read it several times so that you can understand it very clearly. Presynaptic muscarinic receptors are also found on the cholinergic, what you call as the cholinergic synapse, and the output of acetylcholine is thus decreased with repetitive stimulation. So there are presynaptic muscarinic receptors found on the cholinergic nervous system, and these receptors mediate the inhibition of the evoked release of acetylcholine. The evoked release of acetylcholine is also blocked by the inhibitors of acetylcholine esterase and is enhanced by muscarinic receptor antagonists. So, what here it is both nervous systems are being discussed here. The presynaptic muscarinic receptors also influence the release of the norepinephrine from adrenergic neurons in the myocardium, especially in case of the myocardium and vasculature. They are not detected in somatic motor nerves. Presynaptic nicotinic receptors in motor neurons, when stimulated, typically enhance the transmitter release. So this is the action. Of course, how the different type of the mechanisms are brought out by stimulating the presynaptic receptors. Here you can see. Here you can see this membrane potential is always, it is in the negative side. Resting membrane potential is in the negative side, minus 70 millivolts, and this is action potential is arriving here. Whenever action potential arrives, there will be inward movement of the sodium through the sodium channels which are present in the exoplasm. So inward movement of the sodium channel. Then because of this inward movement of the sodium channel, there is intracellular positivity increase which activates the calcium channels. Once again, the calcium channels are also going to open. Then the maximum, what you call as the threshold is reached, then there will be generation of the depolarization process, depolarization, then the potassium channel opens here inside the outside to inside. So as the potassium, potassium channels are going to be increase the polarity or negativity inside, then the repolarization starts. Repolarization start. So because of this thing, particular thing, excitatory postsynaptic produce is produced. Excitatory postsynaptic potential is initiated. Then because of this thing, the action potential is propagated. Whereas, so in the how the inhibitory input is being obtained is by the inward momentum of the potassium channel. The action potential is going to be inhibitory postsynaptic potential, and the chloride ions are coming inside. Chloride ions are coming inside, and potassium ions are going outside. This is known as the inhibitory postsynaptic potential, and the action potential is propagated here, whereas it is inhibited because in the first case, excitatory input, the sodium channels are coming inside. Because of this thing, the action potential is generated, whereas here the chloride ion channels are involved, especially in the IPSP, membrane potential become hyperpolarized, and inhibition of the, what you call as, the action potential. And the materials involved here are the docking complexes like neurexin, syntexin, RAB3, synaptobrevin, and synaptotagmin. This is known as the docking complex. So whenever if you are asked about what is a docking complex, it's a complex containing neurexin, syntexin, RAB3, synaptobrevin, and synaptotagmin. And these are going to fuse the vehicles like this. These are going to fuse the vehicle, and exocytosis occurs in between the synaptic gap. So this is a neurotransmitter receptors, and usually this is a calcium channel. Whenever the inward movement of the sodium is there, then along with that the calcium channels are also going to open, and this is a voltage sensitive sodium channels. As the voltage fluctuation occurs, then the sodium channel is going to open, and the excitatory synaptic vesicles do contain the neurotransmitter, excitatory and inhibitory also containing certain inhibitory neurotransmitters. So this is a picture noting about the action potential propagation inside a nerve terminal.